Hello and welcome. My name is Sarah Cohen, and I am the head of schools with Eton Arrowsmith School. Thank you for joining us tonight to hear about the research findings from three leading researchers looking into the Arrowsmith program. I would like to start by acknowledging that we are gathered here today at UBC on the traditional, ancestral, and unceded territory of the Musqueam people. Dr. Rose will be our first speaker tonight. Gregory Rose is a professor with appointments in the Department of Anatomy and Physiology at Southern Illinois University of Medicine. His research is neuroscience, where he has long focused on understanding mechanisms of synaptic plasticity, learning, and age-related memory impairments. He has over 125 refereed publications in scientific journals, as well as numerous invited book chapters, and over the years has received millions of dollars from national funding agencies to support his scientific studies. Dr. Rachel Weber is a tenure-track instructor with the Faculty of Education at the University of British Columbia, where she directs the BC School in Applied Child Psychology pre-doctoral internship program and the PRTC Neuropsychology Assessment Clinic. Dr. Weber is a leader in the training of school and applied child psychologists in BC at the master's and doctoral level. Dr. Weber's research interests pertain broadly to culturally and neurodiverse populations, including children and youth who are multilingual, who experience learning challenges, who have sustained concussion, and who have survived pediatric cancer. She is currently working on projects that examine the outcomes of cognitive intervention, evaluate neurocognitive function in students with visual impairments, and observe the benefits of participation in opera training. Maybe she'll give us a demo tonight. She is also developing academic and mental health supports for post-secondary students with disabilities at UBC. Dr. Lara Boyd is a neuroscientist and physical therapist at the University of British Columbia. She is a professor and has held a Canada Research Chair, received a Michael Smith Foundation for Health Career Scientist Award, and has been a Peter Wall Scholar. Dr. Boyd directs the Brain Behavior Lab at the University of British Columbia. Her work is centered on answering the question of what limits and what facilitates stroke recovery. Dr. Boyd also serves as the health research advisor to the Vice President for Research and is the university's delegate to the Canadian Institutes of Health Research. Barbara Arrowsmith Young is also here with us this evening. She is the founder of the Arrowsmith program and has been working to help students with learning challenges since 1977. After our presentation, we will be taking questions. So without further ado, Dr. Rose will begin.
Well, thank you for the, probably overly congratulatory and complimentary introduction. As you can tell, I've kind of been around for a while and have explored many of the avenues about how the brain works, and I'm quite pleased to be here tonight, hopefully to give you some insights into the work that we've been doing, trying to understand better how Arrowsmith training is affecting brain and behavior. So the question, of course, is, what is it that we want to do? And, fundamentally, the idea here is that our goal, the goal of the work of me and my group, and probably Laura and Rachel as well, is to really understand how students benefit from the Arrowsmith program. And the reason that I want to know this is because I really would like to understand whether or not the program as it exists is optimal. I mean, are there ways that we can improve it, or can we actually select various approaches that apply better to particular students? So the approach that we've used is twofold. First of all, where we got started actually was to try and understand what was actually changing in the brains of students that received Arrowsmith training. And the way that we decided to approach that was using a technique called functional magnetic resonance imaging, something that Laura also is. She's actually much more of an expert in than I am. But we were actually had a good situation with respect to a I have to talk into the microphone apparently, so the streaming works better. I had access to the appropriate equipment at a nearby hospital, and a convenient arrangement, and a population of students with complex learning disabilities who actually live at something called the Brehm Preparatory School, which is in Carbondale, where my university is. So our initial goal was simply to start taking a look at the brains of these children and find out first, like, how are they different from typically developing controls that we recruited from the local high schools, and secondly, what happened as a consequence of their being in the Arrowsmith program. Secondly, after we had begun our project with imaging, I realized that what we needed to do was to actually better link anything we saw in the brain with changes in behavior. And so this actually turns out to be a much more difficult thing to understand than the imaging from my perspective. What I thought would be appropriate would be to try and use what are considered to be standardized neuropsychological tests. And so we initially targeted areas, as you'll see, that we thought would be appropriate, where we kind of guessed there might be problems with our Arrowsmith population. And later on, when we found that we weren't able to really just bowl a strike and pick a single area that was responsible for the learning challenges that these children had, we broadened our approach. And you'll see what the results are like. And I think the thing to remember is that this is all still a work in progress. I'm happy to be able to say that we've made, I think, some fundamental discoveries along the way. But like most research, it just never ends. So again, the idea was to define the differences between the students that were enrolled in the Arrowsmith program and typically developing controls. And then secondly, to say, well, okay, what is Arrowsmith doing to these students? I mean, there was a lot of information that we were getting, beneficial or positive feedback from both the students and their parents. But could we turn that into something quantifiable, into numbers, into something that would let us better understand how we could improve the program if that was possible? So the first goal was to really try and figure out, well, how are Arrowsmith students different from the typically developing controls? And as I mentioned, and we'll go into detail here and beginning in the next slide, the focus was to use functional magnetic resonance imaging. And there are a number of approaches that one can use here. I chose to look at something called resting state networks. And this will be something I'll define in a minute. The idea was to be able to look at the brain's capacity to function across a wide variety of conditions. And that, I thought that the network approach, which essentially tells you if the brain, how a system is working that would engage in different kinds of behaviors, I felt that that would actually provide us broader and potentially more applicable information than just looking at the brain functioning in a single specific task. When it came to the neuropsychological evaluation, I had talked to many of the students, and we kind of had these ideas that, well, are the students having difficulties because they have like emotional problems or behavioral problems? So we tried to assess this using a standardized test called the BASC, which Dr. Weber probably will tell you more about specifically. We assessed working memory. It's kind of like, can you hold information in your brain and manipulate it and then have some kind of output? There we used something called the Wechsler Intelligence Scale. Executive function, which is considered to be really a key area of challenge for all of us. This is where you actually have to plan to execute things. You have to be able to respond to specific types of information. We thought that this kind of processing deficit, which is where input leads to output, that would potentially be critical for differentiating between a challenge student in the Arrowsmith program and a typically developing control. And finally, toward the end, we decided that, well, we maybe these systems aren't really as affected as we thought they were, but it's a matter of throughput. So, which is kind of given the psychological parlance of speed of processing, that basically I can say something to you, but then it takes you so long to sort of register what I've said and formulate a response that, say, the conversation has moved on, say, especially in a group environment. So then you're kind of left out of it. And so what do? You can't just say, at least more than once, oh, could you please repeat that? Instead, especially if you're a high schooler, which are actually all the students that we looked at, you might, just walk away or, do something spectacular to disrupt the whole conversation. You're just looking for a compensatory mechanism. So, so the idea was, well, okay, maybe they're really, don't have problems with, emotion or working memory or executive process or function, but it's just that they aren't processing at the same speed, and so they're falling behind, and then they have these compensatory behaviors. So these were all of my relatively naive ideas about how one should approach the understanding at a behavioral level what was different between Arrowsmith students and typically developing controls. Well, so let me, with a few slides, first run through the basics of brain imaging and tell you a little bit about what we found. So people have actually looked for a long time for a way to understand how they could measure that the brain was thinking. And this search sort of emerged in the late 19th century when William James, who was like one of the first psychologists, was trying to to quantify the principles of psychology and provide evidence that different functions were taking place at different times. So there was an Italian physiologist named Angelo Mosso who decided that he could maybe measure the brain thinking by putting the subject, and I don't know if I'm going to be able to make this work. Okay, the subject is lays down on a table, which you can see up here. Very delicate balance, and so the patients lying there are actually, in this case, the research subject, and then he would have them think of something. And the idea was that, okay, when the subject started to think of something, there would be like activity in the brain, and that would lead to more blood flow in the brain, and that would make the head heavier, and that would actually make the seesaw tilt toward the head. And in fact, that was something that he saw. Well, so basically, maybe we could argue that this was the first evidence of, of a brain actually thinking, but of course it wasn't very specific. So what's happened, like 100 years, in the 100 years since? Well, in the 100 years since, what we've done, what not me personally, but scientists, mostly physicists, have developed methods that provide a much higher degree of specificity, but rely on the same principle. And that's shown here, that essentially now, instead of having a seesaw, the subject or patient, lies down on a table, and maybe many of you, at least some of you, have had an MRI already. So you lie down and you go in this donut shape, and essentially strong magnetic fields are used to sort of alter some of the ions or molecules that are in your brain, in particular in your blood, and these can be measured. So what happens is that when your neurons, okay, the functional units of your brain are working, well, they require a lot of energy. And so that energy requires oxygen. And so what happens is that then somehow, through a process that we really still don't understand well, the neurons are working and they sort of signal, oh, we need more oxygen. And so as you can see from the cartoon down here, the working neuron then somehow leads to what's called a hemodynamic response, where there's increased blood flow, and in particular, increased flow of oxygen, of oxygenated blood. And it turns out that this oxygenated blood can be can be measured, okay, via the magnetic field. And so, here this kind of shows the overall notion that, you've got some increased neuronal activity that leads to the increased blood flow that leads to something that can be detected by the MRI scanner. This is a technology that actually is relatively new. The first demonstration of this published was in 1992 by a scientist from Bell Labs. So, really only, like not quite 30 years ago. And here, what he was looking at was something very simple. What happens if the subject is in the scanner and you shine a light in their eyes? And you can see that if you were actually recording or looking at the signal in an area that we already knew from anatomical studies was involved in visual processing, you can see that every time the light went on, there was a small signal that was off, the signal goes back down. That if you got progressively further away, there wasn't a signal at all. So instead of saying, well, oh, it's somewhere in the head, now suddenly one can see this isn't the place, but one can see using processing and false color maps that you can actually localize relatively precisely where in the brain, something is happening as a consequence of a behavioral. So it was really only a few years after these initial task-based signals were detected. So task-based meaning that there was you actually gave a discrete stimulus that you understood and had control over, and this evoked a response from some brain region for people to say, well, it's kind of funny. The signals that we're getting are so small. How come they're so small? And this was really a conundrum because the brain, as relatively as small a part of the body as it is, uses a lot of our energy. A disproportionate amount. I mean, 10 times more than you might expect based on its size, and further than one if you're recording signals from the brain, and you're in a the subject is performing a particular task, the change in the signal is really relatively small. And what we envision, it's kind of like when you turn your vacuum cleaner on or off. Okay, when it's off, nothing's happening, and then there's a big change in the signal, and when you turn it on, but not when we record in the brain. So that led some scientists to begin to think, well, maybe the brain is really on all the time. And this was first demonstrated by a scientist named Biswal. And what he was doing was actually just recording the signal fluctuation, so the bold fluctuation over time. And so instead of looking at a signal that was maybe in fractions of a second or sections, you can see that here a long time, and you can see up and down and up and down all the time. So this was the idea that there were actually changes in signal strength that happened during rest. And further, what he found was that these really low frequency oscillations occurring very slowly, that they didn't happen unique, that these fluctuations weren't unique to a single brain region, but in fact, certain brain regions were kind of like synchronized. I mean, not necessarily directly wired together, but that their signals, their bold signals, moved in concert. And so this is the idea that by looking around the brain and sort of saying, well, which areas seem to be fluctuating at the same, sort of the same way at the same time, that these things called resting state networks were discovered. And so here we can see this is this is a view from the top. So imagine, you cut off the skull and you're looking at you. Well, what was the value of this? At the time, people then began to speculate, well, I know that these are brain regions that are active when you actually ask the subject to say make a movement or even to visualize a movement. um, could we use these signals to kind of make predictions about the capacity of the brain to act when it needs to? And that's the fundamental notion behind the idea of resting state networks. So I guess I went the wrong direction there. So since 1995, the number of networks that have been discovered and described really has exploded. And right now we're saying, okay, well, maybe there are a dozen, and this is very arguable. I mean, some people have actually used sort of microanatomical mapping to say, oh, well, there are hundreds of networks. And our problem is that you start to have so many units of anything that you just can't understand what they do. So I kind of like the idea of the initial description that kind of maxed out at maybe a dozen networks at the in the late 2000s. Well, some of these make perfect sense. Like there are areas in the back of the brain that are responsible for visual processing. There are areas on the side of the brain that are responsible for auditory processing. We've already seen this sensory motor network. Well, I think the networks that are potentially a lot more interesting are the ones that we think are involved in thinking. Okay, and so again, here's where we have a cross section. So this are not necessarily related to action, but are in the middle between receiving stimuli and figuring out what you're going to do about them. Now, the other thing that was identified relatively early on in this process was that while there are brain regions that kind of coordinate their activity, there are other regions that basically turn off when a particular network is turned on. So what one can see from this diagram over here is that yellow and the orange, okay, are really pretty much cycling together. But then the blue so it leads to the idea that, in fact, there's some sort of control of networks, and that basically, under the appropriate behavioral conditions, if one network turns on, then another network turns off. So, this, of course, leads to the idea that there's really exquisite control and, a tremendous amount of information processing that can happen through these resting state networks. So why have I chosen to focus on resting state networks rather than simply, task-based, more traditional fMRI methods? Well, the main reason, as I mentioned at the beginning, was because I think that they actually give me more analytical power. Now, practically, it's a lot simpler because you really only need a scanner. And so you don't need to have, a monitor and a response pad and lots of maybe training in a task and all the things that you need if you're going to do task-based fMRI. Not that this is not also a very useful thing, but I'm trying to paint initially with a broad brush. I think it's something that is a lot easier to standardize across sites. So you don't have to worry. Like a lot of my career has come from doing what's called preclinical work. So you're looking at behavioral studies in rodents, and 3 labs will do ostensibly the same study, and they'll get different results. Well, so either nothing is replicable, which is something that some people would argue, or you could say, well, the problem is that the studies, they say they're being done the same way, but they're really not. And I wasn't interested in kind of going down that road again with human studies. So here, the subject lies in the scanner and basically just thinks about whatever they want. What you don't want is you don't want them to think about something that's going to make them really angry, and you don't want them to fall asleep. But that's basically it. I think a big advantage also of looking at the resting state networks is that you can get the data and assess multiple networks at the same time. If I give you a visual processing task, then okay, I'll look in auditory or in visual cortex, and be able to tell you something about how that particular brain region works. But I can't tell you anything about how the auditory cortex that processes hearing works. And when we still, our understanding of the cognitive functions of the brain are still so rudimentary, I think the idea of being able to gather a lot of data at once and to, potentially pour back over it as we understand better about what the brain is doing is a real advantage. I mean, once we have these data, we can spend years analyzing it if we want to. The A general problem with neuropsychological testing is that there's something called a practice effect, and so it's kind of like, well, the first time you do something, you're not very good at it, but the more you practice it, the better you get. And so this can be really a problem if you want to do repeated assessments. For example, say, okay, well, so what's the benefit of Arrowsmith training? And so you can get a baseline and then test the same subject again, say a couple weeks or a month later, and they say, oh yeah, well, they're really better at this task. But the question is, why are they better? Are they better just because this is the second time around, or is it because they actually, something in their brain has changed? So the thing that theoretically is an advantage of the resting state networks is they can be done frequently, and we already know that that it is a stable measure, so that if things don't change, it doesn't change. And then finally, it has actually predictive power, and this is an evolving area. But I put up some examples here looking at some of the different networks, which I'll explain more in a minute. But you can see that there is a relationship in the connectivity of the strength, or in this case, okay, so basically, while this literature is really still developing, the idea is that you can actually measure the strength of a resting state network, and that will allow you to predict how well the subject will do subsequently in a behavioral task. So there are all these things that I think, make a good case for at least starting out with this. Well, so if there are, come on, so many networks, where are we going to start? Our idea was to just focus our attention on 4 networks that we think are central to this process of dealing with new information. And so these are the default mode network, the dorsal attention network, the salience network, and the frontoparietal control network, or sometimes called the executive control network. And in each case, these are networks that many labs have identified. They're very stable in terms of the brain regions that are put together to make these networks. And they all seem to be involved in like different aspects of, thought processes, from registration to generation of action. And so what I've done here is to, kind of make you a little capsule. Okay, the default mode network most active when you are not engaged in an action. Okay, it's kind of like when you're tuning out the outside world and you're thinking about it. So, it's sort of a two-state universe where either you're engaged in the outside world, you're acquiring information, or you've disengaged from the outside world and you're just thinking about it, you're processing it. And, I think all of us are sort of just generally aware of this idea that, well, okay, you're paying attention to something, or no, you're not really paying attention to something outside, you're paying attention to something inside. What is that voice inside your head telling you? Well, so we've got the default mode that then once you're engaged in an action tends to turn off. And there are, I think, 2 critical steps in terms of modulating what you're attending to. So there's the dorsal attention network that just sort of recognizes that there's a lot going on in the outside world. And perhaps more importantly, there's something called the salience network. So with all the things going on in the outside world, how do you decide what it is you want to pay attention to? You know, some of you may be aware that you can see flashing lights going by outside because there are cars on the street and it's getting to be dusk. But, you know, you may be aware of that. Your attention network help you to see that. But your salience network says, well, you know, okay, that's not important for me right now. What's important for me right now is to understand what Dr. Rose is saying so I can ask him a hard question later on. Okay, so that's kind of the basic idea. And then of course you need to plan an action after you've, you know, decided what it is you want to do, and that's where the planning center is really within this frontoparietal control network. So we were interested in assessing the basically the baseline functions of these four networks, not only how strong are the network connections within themselves, but what about the connections between the networks? Because the networks really, we feel, need to work together. And as I presented some information showing that they can work together, that one network turns on, another one turns off. So somehow they're modulating each other, and that very likely is necessary for the brain to work in an optimal fashion. So what do we see if we look at the network connectivity in Arrowsmith students? Okay, and so what this figure shows, we're just looking at network nodes here, not at everything in the brain. And there are two new views for you to absorb. Over here on the left, and can I take this thing? Okay, so now I won't be forced to come back here to the. All right, here on the left, once again, we have, you know, what's called the transverse section of the horizontal section. So we've cut off the top of the brain. We're looking down from the top. And over here, we have something that's called a connectome. And it's the same information, but to me, often this gets so confusing. And, you know, especially if there are a bunch of nodes, you know, I can't really figure out what is going on. So if I want to look at connections, what this does is sort of bring them out into a ring. Okay, and that just makes it easier to visualize. So we're seeing a couple of things. We're seeing, first of all, that warm colors like red mean that the connections are stronger, and blue colors mean that they're weaker. And so you'd say, well, stronger or weaker with respect to what? Well, what this is, it's an analysis that directly compares the Arrowsmith students to the typically developing high schoolers. So if you subtract the high schoolers, the typically developing high schoolers, from the Arrowsmith students, what you can see is that there's a lot of activity in the brain that is actually stronger in the Arrowsmith students than it is in the typically developing students, which to me, at least initially, was a little bit of a surprise. However, there are some connections that are actually not as strong, the blue ones, in the Arrowsmith students. And it turns out that those connections actually are largely restricted to the salience network. Okay, so what we seem to be seeing here, first of all, that the brains of the Arrowsmith students are kind of working harder at rest than the typically developing controls, but that basically the network that's supposed to be responsible for figuring out, of all the things that you are in your environment that you've noticed, what should you be paying attention to, what's important, that those connections are actually weaker in the Arrowsmith students than they are in the typically developing controls. So what we found, and you know, you sort of can formalize this, is through the term hyperconnectivity. And so here, what we've looked at is we've actually quantified, I mean, we've measured the strength of the connections both within the networks, because remember, each network has a bunch of different sites, and it's like, how well are they connected together, as well as between the networks. So, you know, how strong is the connection from, for example, the default mode network to any of the other networks? And what you can see over here on the left is that the Arrowsmith students really have pretty much the same within network connectivity as the typically developing students. It's maybe a little bit less, but not enough to reach statistical significance, which is what we use as kind of a baseline measure for deciding something is important, and it's not just biological noise. On the other hand, if you look at the between network connectivity, you can see that the Arrowsmith students are much more connected. A given network is much more connected to other networks. And in this case, what I've done is to mark each individual network with an asterisk, which is a standard sort of way of demonstrating or indicating statistical significance um, between the control and the Arrowsmith group. And so basically, all these connections are hyper connected. They're overly connected with other networks. And it just turns out that the frontoparietal control network, there's enough variability in it that with our sample, it didn't reach statistical significance. But you can still see that there's, a big change. Well, so we've got this difference between typically developing control students and Arrowsmith students, which I guess that is reinforcing, because you'd say, well, at a behavioral level, there's a reason why my child is at Arrowsmith. It would be disappointing, or it would certainly not validate the method we're using if with the functional MRI we didn't see any differences between Arrowsmith students and others. So, of course, the next question to explore is, well, what's the effect of Arrowsmith training? And, this turns out to be a somewhat complicated question, because the nature of the program is such that each student is initially evaluated and then gets a customized program. So conveniently, it turns out that they have a summer program where all students are trained on the same single task, and they have an intensive training period. So from the standpoint of initially designing an experiment, this was really beneficial for us. And so what we did was to begin our analysis by looking at students that had participated in this summer intensive program, the cognitive intensive program. They're trained in what's called the symbol relations task, which probably all of you know even better than I. Very intensive training. The task has progressive difficulty, so it's like really hard to get to the end. They have to have a high level of accuracy at any given level before they advance to the next level. So the students work really hard on this, and the idea, I think it was designed really to sort of try and compress, say, a year's worth of training into 6 weeks. So you can imagine, I mean, this is really, I'm impressed that the kids are willing to put up with this, but they do, and they make considerable progress, as you'll see. So here are group data just showing the change in behavior. And so what you can see is that at the start, using our quantification scheme, that the students really are all performing fairly poorly. I mean, they can do the task, but they don't get very far in this scenario. But after they finish training, I mean, their performance has improved like, 3 or 4-fold. Further, what is actually very useful for us from the experimental standpoint, looking over here on the right, you can see that students start at a different place and that they finish at a different place. So there's a lot of individual variability in performance. And what that allows us to do is to actually be able to correlate changes in the strength of the networks with individual changes in performance. So here's what it looks like if we look at the networks again and we say, well, okay, what has happened as a function of this training in the symbol relations task and the cognitive intensive program? And what you can see is that there are changes in strength in a number of connections, and over here shown in the connectome. But what's really interesting about this is that actually most of the changes are involving sites within the salience network. So remember, the salience network is the one network that really was weakened in the Arrowsmith students compared to the typically developing controls. So this single task seems to really focus itself on kind of working in the direction of normalizing the strength of this network. So this was the data that we saw with the entire correlation analysis, and I wanted to just kind of take a look at this in a little bit more detail. And the way I did that was comparing two groups of students that were all trained at the same time. So basically, all of the students that are included in this set, which is a relatively small data set because we only had a total of 16 students that we could use the scanning from, but I just took the 4 students that improved the least and compared them to the 4 students that improved the most. So you can see over here that, the worst performing students really barely improved. They started out with a score of 2 and they made it up on average to a score of 3. So, hardly improved. Whereas the best students started out at a score of 2 and ended up at a score, actually, they ended up at a score of 11. So they improved by a factor of, not a factor, but a number of 9. So they really were doing incredibly well. Um, Astoundingly well. I don't think I could have improved that much over a 6-week training period. So then we just looked at the network connections and we said, well, okay, so what's happened? We already know that overall, proportional to the training, that we see these changes, a strengthening of network connections. Well, if you don't improve very much, it turns out nothing changes. I mean, we're not seeing any strengthening or weakening. We're just seeing nothing. Whereas if we look at the ones that improved the most, okay, then we are seeing, again, strengthening and a lot of components that are involved in the salience network. So to me, this actually really reinforced the idea that, number one, changes in behavior were associated in changes in brain function. And number two, that really an important player of the networks we looked at is the salience network. So this was after, like looking at this and sort of gaining a set of expectations. Remember, we start out and we say, well, at baseline, the Arrowsmith students are hyper connected. Okay, that basically their network connections are stronger. The connections between the networks are stronger in the Arrowsmith students than they are in the typically developing controls. Maybe the opposite of what you might expect, but it's easy to rationalize and saying, well, gee, maybe all these connections are working harder because they're missing a key piece of information, like from the salience network, about what they should be paying attention to. So they're just like always in this hyped up state, just waiting for information. If we look at the consequences of training in the Arrowsmith program for 1 year, okay, and so this is this quite varied, individualized curriculum, what do we see? And what I did here was to analyze a group of subjects that received Arrowsmith training along with the standardized BREM curriculum in Carbondale, where I live, versus students that received the Arrowsmith curriculum, which is not the Arrowsmith component is not different, but the academic component is a bit different up here in Toronto. And if you look up here on these two connectome rings, you can see that fundamentally you're seeing the same thing. So what is it that you're seeing? Well, you're seeing a lot of blue, okay? You're seeing a lot of blue with a little bit of orange, okay? So a little bit of strengthening and a lot of weakening, okay? And this I thought was reinforcing given where we are at the moment, because remember, I'm telling you that at baseline, the Arrowsmith students' brains through these networks are working harder. One could even argue that they're working overtime. But after they've been in the Arrowsmith program for a year, these things have all toned down. So these are basically showing what is the comparison between the baseline Arrowsmith scan and the scan at the end of the academic year. So at the end of the academic year, the between network connections are not as strong as they used to be. They've toned down. And this toning down is exactly what you would expect if the brain function was being normalized, right? You've got a hyperconnected brain, you want to unconnect it. And I'm not saying that this solves all the problems of a student, but I think for an initial finding, it's really provocative. Number one, that we've got this issue of the salience network in particular that is underperforming, okay, relative to a typically developing control. And so the rest of the networks are all kind of like hyperactive and interacting, to too great a degree with each other. And that secondarily, after the Arrowsmith training, that all these other connections have kind of like toned down, that they've moved in a normal direction. So that's what we have seen so far with our imaging studies. And I think it really provides a basis for us to come up with a whole bunch of new ideas and new hypotheses, experiments for us to perform to try and better understand how the Arrowsmith program is actually changing the brain. And I think it's reasonable to argue, well, these are changes in brain. And, I think each one of us may have a different definition of neuroplasticity, but in general, this certainly falls under the general category of a plastic change. I mean, the brain is working differently. So what we were then, I sort of decided that it was important for us to try and tie this to something that made more sense. I mean, basically, you guys want to have your child performing better in school. You want to have their behavior be better. What can we begin to link these changes that we're seeing in the brain imaging to, sort of functional behavioral real world changes? And this is a process that we're still working on, I have to say. What we found was that, there are lots of changes between or differences between the Arrowsmith students and our typically developing controls. They were, impaired in our groups in working memory and executive function, in areas that we initially guessed there might be a problem. In our cohort, we did not see any differences in social emotional functioning. And you'll see Dr. Weber will show data from her population. It turns out that again, this is probably at the moment just differences in the populations, because our baseline scores, our kids scored a lot more normally, I guess you would say, in the social emotional measures. So, there just may not have been as much room to improve. And so it isn't to say that Arrowsmith training doesn't improve those. And for individual students that are in a deficit condition, it could be quite beneficial. But on average, I looked at this quite carefully after seeing the data that had come from Vancouver, and no, we still don't have any basis for change there. So in Carbondale, we were, sort of like taking single shots from our rifle. And so one year we would look at executive function, another year we might look at speed of processing. Every year we kind of do a different thing. We had a small sample size. And what we found that was kind of confusing was that, not only did we not see changes in a number of measures where we guessed there would be a change, but even in tasks that were supposed to assess the same thing, we really would see, we would see a difference with Arrowsmith training in some tasks, but not others. And in particular, there is one kind of complicated task called the tower task, which is part of the DCAPS battery, which is all supposed to be an executive function assessment, that showed a significant improvement after the year of Arrowsmith training, but nothing else. And so I was kind of frustrated by that, in particular, since I'm not an expert in neuropsychological testing. So with the Toronto group, we tried to really begin a little more of a shotgun approach, if you will, where we had a licensed clinical neuropsychologist who could deliver many tests from a standardized battery. So, the group kind of got together and said, well, what should we take a look at? And again, we're looking at, where do we start here, and where might we see a potential change with the Arrowsmith training? Well, so this is a complicated slide. And this shows these poor kids, all the tests that they underwent, where we really used a broad battery. And simply what you can see are two sort of categories. Okay, number one, and all the blue bars are the baseline scores. So when the kids were assessed at the beginning of the academic year, the orange bars are at the end of the year. All right, so beginning and end. Secondarily, using this Woodcock-Johnson battery, they actually divide their tests into 3 domains. Okay, oral, cognitive, and achievement. Achievement are supposed to be actually, they supposedly predict academic performance, even though I'm not convinced of that quite yet. So over here, everything under the blue line is the cognitive. These are individual cognitive tests. Some of them actually are composites that are where the values from individual tests are weighted and put together. Over here are the achievement tests, and then this one is a special test where this was part of our idea about speed of processing. In the different versions of the Woodcock, in version 3, there's something called decision speed. We ran that in Carbondale and didn't see any change. And I thought, well, this is ridiculous. There has to be a change. So we included it here. And you can see that again with the Toronto group, which actually was a larger sample size, once again, we didn't see any change. Well, so the asterisks indicate individual tests for which we saw statistically significant improvement after a year of Arrowsmith training. Okay, these are all in the Toronto group, so they're all assessed by the same person. So, we eliminate a lot of the variability. And you can see that really there's quite a number of tests that show statistically significant improvement as a function of Arrowsmith training. I mean, to my mind, a surprising number of tests, perhaps not to Barbara, but to me. And so overall, I mean, the overall improvement was highly significant. Not in everything. Short-term working memory, for example, we didn't see a change. And that's what it looks like on the graph. But many other things. And then basically on the right here with the achievement scores, you can see that actually measures like academic fluency and math facts fluency, everything actually that we measured except for the sentence reading fluency showed a statistically significant difference. Now these are expressed as the W score, and the W score is a proprietary measure that the Woodcock-Johnson provides as a calculation based on the data you give it, and they claim that this is a score that actually is very useful because it more accurately identifies longitudinal changes over time. So, the scale is supposed to be linear wherever you are. And, then theoretically, this is also helpful in terms of identifying whether or not these are actually going to be meaningful changes with respect to a student's academic performance. So, the long and the short of it was that we actually saw quite a number of changes that seemed to be moving in a positive direction. So the question then was, well, could we see any kinds of changes, in sort of the grouped curriculum, the mass trials curriculum of the cognitive intensive program training? So last summer, this is what we did. We gave a subset of these tests, and what you can see in this case is that once again, there are a number of significant changes on the cognitive side. But interestingly enough, with the 6-week battery, we saw nothing on the academic side or on the achievement side. And, the argument that could be made here is that, well, either it takes longer for these changes in the toolbox and basic skills to translate into measurable changes in academic performance, or my pet idea that hopefully we'll explore someday is that, in fact, during the regular year curriculum, the students actually do receive academic instruction along with all of the individualized measures with the Arrowsmith toolbox. So, I think basically it could be that having these things being delivered together is more beneficial to the brain than having them be apart. But the main point is, you can see, number one, that even the short trial or short training period still produces change in, what are considered by the community, the psychological community, to be validated neuropsychological measures. So this is where we actually are at the moment. And, I think hopefully I've been able to convince you that basically the Arrowsmith students are really a heterogeneous group. That they're not performing on any measures that we've looked at so far at the same level as the typically developing controls, except in our social emotional function. But, again, I don't know if that's the difference in our cohort or sensitivity of the test or whatever. We know that, that the performance in the neuropsychological tests and, of course, in academic performance is paralleled by changes in network connectivity. And that paradoxically, at least based on our expectation, where you might have said, oh, the brain of an Arrowsmith student, it's just not working as hard. Well, based on these resting state network analysis, in fact, it seems to be working harder. they are hyper connected. There's more connection than there should be, and that's the paradox where we wouldn't have thought that would be the thing we would observe, and that but the exception to that was that the connections within the salience network were actually weaker. And so, this of course leads to the idea that, well, the salience network may be controlling the other, the subsequent function of the connectivity between the other networks. And that's something that we need to study and explore. We also showed, I think very importantly, that we have the tools to demonstrate that Arrowsmith training does something, that we simply have the sensitivity to do that. And, at the beginning, of course, we might have had a hypothesis that based on, reports from parents, reports from students, that things would get better. Well, the main point was that we actually saw changes. And then our goal, of course, is to try eventually put a story together that says, well, okay, if behavior is better, what is changing in the brain, and how is that reflected in these, formalized neuropsychological tests that theoretically are telling us more specifically about the functions of the brain? How does all of this come together? And what we learned from the SIP data, the cognitive intensive program, was that we saw changes in the network connectivity that was proportional to the amount of improvement that the students showed over the training interval. So I think it makes it a lot more believable, not simply that there was an on-off switch, but that the degree of change in an individual brain was reflected in a change in the strength of their network connections. And actually the network that seemed to really emerge from this was the salience network, or the network that helps you to decide what it is in your environment that you detected is important, that you should be paying attention to at a given moment. We also learned that, training over a year's academic period generally seemed to tone down this hyperconnectivity that we saw within the Arrowsmith student population. And then, as I've shown you from our initial forays into the neuropsychological testing field, trying to provide a bridge between brain and behavior, that we, saw parallel improvements in many, many neuropsychological measures, both indicators of cognitive function and hopefully measures that predict improved academic achievement. So that's all I have to say for the moment. And should we take questions, or Rachel, do you want to talk and then we just take all the questions at the end? Well, I'm not sure how you want to proceed here. Okay, so what we're going to do is save questions at the end, because then it'll be, you'll have had a chance to see, kind of like everything that all of us have to show, and that might help you, decide like which questions are more important than others. So with that, I believe that it looks like, is it Laura or is it Rachel? Oh, okay, fine. Yeah. Well, let's first see, are we advancing? Yeah, great.